This video tutorial is all about the use of biotechnology to make food. Microorganisms have been used for many years, and many traditional foods are made with the help of microbes. For example, yogurt. Yogurt is milk that has undergone fermentation by either lactobacillus bacteria or streptococcus bacteria. The bacteria convert lactose into lactic acid. The acidity denatures the milk protein, causing it to coagulate. The bacteria partially digest the milk, making it easier for us to digest. Fermentation also produces the flavors characteristic of yogurt. Other bacteria can be added, such as probiotics, which make or may benefit the human health by improving digestion of lactose, aiding gastrointestinal function and stimulating our immune system. Secondly, cheese. Milk is usually pre-treated with a culture of bacteria, for example, lactobacillus, that can produce lactic acid from lactose, as we saw in yogurt production. Once it is acidified, the milk is mixed with rennet. Rennet contains the enzyme rennin, which is found in the stomachs of young mammals. Rennin coagulates the milk protein in the presence of calcium ions. The resulting solid, called curd, is separated from the liquid component, called whey, by cutting, stirring, and heating. The bacteria continue to grow, producing more lactic acid, and the curd is then pressed into molds. Treatment whilst making and pressing the curd determines the characteristic of the cheese. Flavour is determined using either other bacteria or fungi that can be added and sort of happens during the later ripening and maturation stages. Thirdly, bread. In bread production, we use yeast, which is a single-celled fungus. The yeast undergoes fermentation during the proving stage. It respires anaerobically, producing carbon dioxide gas, which enables the dough to rise. We also use yeast in the production of alcohols. For example, in wine, which is made using grapes, there's natural yeast on the skins of the grapes. The grapes contain sugars, specifically kind of like fructose and glucose. And so when the grapes are crushed, the yeast will use these sugars to produce carbon dioxide and alcohol. Ale or beer is brewed using barley grains that are beginning to germinate through a process called malting. As the grain germinates, it converts starch to maltose, which is respired by yeast. Anaerobic respiration produces carbon dioxide and alcohol, and the hops are then used to give it that bitter taste to the liquid. More recently, microorganisms have been used to manufacture protein that is used directly as food. The microorganism used most frequently is a fungus. The fungal protein, or mycoprotein, is also known as a single-cell protein, SCP. The best known example of mycoprotein is corn, which was first produced in the early 1980s. It is marketed as a meat substitute for vegetarians or a healthy option for non-vegetarians, as it contains no animal fat or cholesterol. There is huge potential for single-celled protein production using such microorganisms as candidinae. I can't really pronounce that very well, but that is a fungi, which can produce protein with a similar amino acid protein profile to animals and plant proteins. These fungi can grow on almost any organic substrate, including waste material such as paper, and whey, which is that curdled milk we were mentioning earlier. There are loads of advantages and disadvantages of using sort of fungi to produce protein. Firstly, the production of protein can be many times faster than that of animal or plant protein. And the biomass produced has a really high protein content, often sort of between 45 to 85%. The production of protein in this way can be increased or decreased according to demand, and there are no animal welfare issues. We're using a fungus here. The microorganisms provide a good source of protein, and the protein contains no animal fat or cholesterol. The microorganisms can be genetically modified to adjust the amino acid content of the protein, and the production is independent of seasonal variation. As well as all of this, not much land is required. However, some people may not want to eat fungal protein or food that has been grown on waste, such as paper or whey. There's a little bit of a faff relating to the isolation of the actual protein, because the microorganisms are grown in these sort of huge fermenters and need to be isolated from the material on which they've grown. The protein has to also be purified to ensure it's not contaminated. The amino acid profile may be different from traditional animal proteins, so it might be deficient in certain amino acids. And there's the risk of infection. So the conditions needed for the microorganisms to grow are also ideal for pathogenic organisms. So care must be taken to ensure the culture is not infected with the wrong organisms. And finally, palatability. The protein does not always have the taste or texture of sort of traditional protein sources, which is another reason why some people don't opt for it.